Is SARS-CoV-2, the novel coronavirus that causes COVID-19, not a truly isolated human virus, because it was only replicated in monkey cell lines? No, that's not true. While the origin of the virus is hypothesized to be from an animal, the virus was isolated from human patients soon after its emergence in humans. SARS-CoV-2 replication in human cell lines has also been observed. The claims appeared in an article, published by Natural News on August 11, 2021. It cited a video interview with Judy Mikovits, a virologist and anti-vaccine conspiracy theorist who has made debunked claims about COVID. A section of the article reads, quote, "Every biosafety level 4 lab that claimed to isolate SARS-CoV-2 from humans ultimately placed the sample in a monkey kidney cell line to prove its replication. This virus is not a truly isolated human virus because it could only be replicated in the monkey kidney cell line. To determine whether SARS-CoV-2 is an infectious human virus, it must be taken from the blood, sputum, or lung lavage of an infected human, but the infectious RNA of SARS-CoV-2 was never cultured in a continuously growing human cell line that supports the replication of SARS-CoV-2. Since this novel coronavirus only replicates demonstrably in vero monkey cell lines, it has never been proven to originate or spread from human to human, let alone spread from healthy people. Since the current flu, MMR and polio vaccines contain these same vero monkey cell lines, the only way to deploy SARS-CoV-2 is through transfection via these needles." End quote. This is how the article appeared online on November 10, 2021. Source: Natural News. Screenshot taken on WED November 10, 22 hours 24 minutes and 50 seconds 2021 UTC. These claims are not true, as explained below. Misinterpreted claim. Every biosafety level 4 lab that claimed to isolate SARS-CoV-2 from humans ultimately placed the sample in a monkey kidney cell line to prove its replication. Lead Stories reached out to the Centers for Disease Control and Prevention, CDC, for more information about SARS-CoV-2 isolation and was referred to the agency's resource, SARS-CoV-2 Viral Culturing at CDC. The resource contains links to the article, Severe Acute Respiratory Syndrome Coronavirus 2 from Patient with Coronavirus Disease, United States. The abstract describes researchers isolating SARS-CoV-2 from samples taken from the first U.S. patient infected with the virus and reads in part, quote, "The etiologic agent of an outbreak of pneumonia in Wuhan, China, was identified as Severe Acute Respiratory Syndrome Coronavirus 2 in January 2020. A patient in the United States was given a diagnosis of infection with this virus by the state of Washington and the U.S. Centers for Disease Control and Prevention on January 20, 2020. We isolated virus from nasopharyngeal and oropharyngeal specimens from this patient and characterized the viral sequence, replication properties, and cell culture tropism." End quote. The isolated virus was placed in both human cell lines and non-human cell lines, meaning it was not exclusively placed in a vero cell line, which is most likely the monkey kidney cell line, the claim references. According to the results section of the article, quote, "Because research has been initiated to study and respond to SARS-CoV-2, information about cell lines and types susceptible to infection is needed. Therefore, we examined the capacity of SARS-CoV-2 to infect and replicate in several common primate and human cell lines, including human adenocarcinoma cells, A549, human liver cells, HA7.0, and human embryonic kidney cells, HEK293T, in addition to vero E6 and vero CCL81 cells. We also examined an available big brown bat kidney cell line, EFK3B, for SARS-CoV-2 replication capacity." End quote. False claim. This virus is not a truly isolated human virus because it could only be replicated in the monkey kidney cell line. The basis of this claim may have originated from the CDC study stating that no CPE was observed in any of the cell lines except in vero cells. However, John Lednicki, a research professor in environmental and global health at the University of Florida who is affiliated with the university's Emerging Pathogens Institute, clarified that a cytopathic effect, the aforementioned CPE, is not the same as viral replication. CPE is defined as a structural change in a cell due to a viral infection. During a phone call with LED Stories on November 10, 2021, Lipnicki said, quote, "There are many viruses that will grow and replicate but not kill cells or not form noticeable cytopathic effects. The presence of cytopathic effects it's not the only criteria that the virus is replicating." End quote. Indeed, the CDC study went on to describe some SARS-CoV-2 replication in human cell lines. Quote, "In contrast, HUH7